Yes, it will be signaling starvation. Okay? Yes?
So is lactose the only sugar that has this?
Lactose, it's also a very good question. Lactose is the only sugar that has this characteristic in the E. coli genome. The only time it's present in the E. coli genome. Yeah. Okay. So, very cool mechanism, very cool control. Now, your book goes to all kinds of details about different positive and negative methods, et cetera, et cetera. That's not what we're going to be so much concerned about. Okay. You can see what happens during repression. I'll just lead you through it. But here is, by the way, I should also mention another term I didn't give you. The place where the repressor binds, okay, that's called the operator. So if it binds to the operator is the thing that operates the operon. So by binding to the operator, it's controlling that control region of the operon and thereby shutting it off. Now, another term to give you, lactose is called an inducer of the operon. And it's called that because when it's present, it induces the transcription of that operon. It does it by binding to the repressor. Okay. Now, the three, yeah. Yes. Sure. So the inducer you're talking about? Yeah. So lactose is called the inducer because it is the molecule that induces the production, the transcription of the operon. Without it, that doesn't happen. Now, what you see here is a little bit of a confusing figure, and I pointed it out to you because you'll see this and you'll wonder what the operon is. The operon is everything to the right of my green pointer, everything over this direction. It has three genes called LAC-Z, LAC-Y, and LAC-A. Okay? And the only one we really need to worry too much about is LAC-Z. LAC-Z catalyzes the breakdown of lactose into glucose and galactose. That's why the cell needs this. If it can't break down lactose to glucose and galactose, it can't use lactose. Okay? That's one consideration. Now, the repressor turns out to be coded by a gene that's right next to the operon. It's not a part of the operon. All right? So the gene that codes for the repressor is called LAC-I. I standing for inhibitor. All right? So you see the gene for the repressor, it's not part of the LAC operon. It has its own promoter. It has its own promoter so that it's going to be made, and it's not going to be affected by the presence or absence of lactose. As I said, the repressor is almost always present in small quantities so that it can always potentially be there to turn off the LAC operon if necessary. Yes?
Well, what exactly did you say LAC-Z catalyzes the breakdown?
So LAC-Z, okay, the protein that's made by that's coded by the LAC-Z gene, catalyzes the breakdown of lactose into glucose and galactose. That's what makes up lactose. Mm-hmm. Okay, so that's the operon itself, and we can see a variety of things there. Let's see, what else did I want to say? We're not going to talk about that, and we're not going to talk about that. Keep it simple, I say. The last one I want to talk about in the prokaryotes is another operon. And this operon, I think students find kind of confusing. So I'm going to try to keep it as simple as I can. All right? This operon that I'm going to describe to you is found for several different operons in E. coli. This one controls how the cell makes tryptophan. E. coli cells have to make their own amino acids. They have to make them from scratch. And it takes quite a few enzymes to do that. In the case of tryptophan, there are 10 different enzymes that are necessary. So this operon has 10 genes on it. It's a long messenger RNA that is made. 10 different genes. Okay? The structure of the operon looks something like this. Okay? A whole bunch of names and letters, and we're not going to worry about the names and letters. 10 genes. We're thinking long messenger RNA. Let's think about the needs of the cell. The cell has lots of tryptophan. Does it want to be making this operon? No. It doesn't need it, right? It's got plenty of tryptophan. It's going to waste energy if it makes 10 different genes to make more tryptophan. So when tryptophan is abundant, the cell doesn't want to be making this operon. Right? What if tryptophan is short? If tryptophan is short, then the cell says, whoa, we better start making some tryptophan, because if we can't make tryptophan, we can't make proteins. So there's two simple situations. Tryptophan abundant, tryptophan not abundant. Okay? Not abundant, we want to have the operon on. Abundant, we want the operon off. All right? Everybody got that? Now, the way in which this operon is controlled is by a mechanism called attenuation. That's got an extra T in it, I just noticed. A-T-T-E-N-U-A-T-I-O-N. That first T is not, or that middle T right there is not correct. Let's look at this unusual thing. Okay? Attenuation is controlled essentially by the abundance or the scarcity of tryptophan. Now, when we look at the tryptophan operon, we see that the tryptophan operon, we're looking now in the messenger RNA that's being made. Okay? We look at the messenger RNA that's being made, and we see that we have the potential to make various base-paired structures. Here's one possibility. Here's another possibility. Same sequence. They're two very different kinds of structures. Okay? Everybody with me so far? So the same sequence, we have two different kinds of structures that can form. One structure, if I look at the one on the left, is going to cause the RNA polymerase to fall apart. It's a terminator sequence, factor independent. The other one is called an anti-terminator, and if it forms, transcription will continue to go. So then the real question is, well, which one forms? This can form or this can form. Does this cell flip a coin? Well, that wouldn't be a very good way of controlling things. We'd like to know more about what determines which one forms. To understand this, we have to understand how translation occurs in bacteria, because translation plays a role in this. We haven't talked about translation. Translation, of course, is how the ribosome moves along the messenger RNA and starts making protein. Okay? So here's a messenger RNA, and the ribosome would normally come along here and say, oh, here's a sequence I can start translating. I'm going to start making protein. Everybody with me? Now, notice that this little sequence, and we're very close to the end. That is the very beginning of the messenger RNA. We're very close to the beginning. All right? There's position 50. We've only gone 50 nucleotides in. This guy might have 10,000 nucleotides if it goes full length. So we're very early. The ribosome is coming along, it starts translating, and it gets down to here, and notice we've got sequences that specify here is where a tryptophan should go, here is where a tryptophan should go. So in building this protein that's coded by this guy has two tryptophans very early. You with me so far? Ribosome's sitting there. The ribosome gets to here and says, oh, it's reading the genetic code, I've got to put a tryptophan in here. The ribosome looks around, and there's no tryptophan. Okay? Or the ribosome looks around and it says, there's a lot of tryptophan. If there's a lot of tryptophan, the ribosome says, oh, grab some tryptophan. Okay? We're going to grab tryptophan. We are going to put it into this protein that we're making, and we're going to move along our merry way. Okay? If there's no tryptophan, the ribosome is going to sit here and it's going to look and it's going to wait for some tryptophan and wait for some tryptophan and wait for some tryptophan, and finally, it's so scarce, a little bit shows up, and it says it finally makes its way through and moves on to the next amino acids to put in. Everybody with me so far? Two situations. A lot of tryptophan, ribosome just moves through very quickly. If there's little tryptophan, the ribosome sits here on the same sequence, here's that sequence right here, and waits for tryptophan to come. Ribosome is moving fast, ribosome is moving slow. Moving slow when there's no tryptophan, moving fast when there is tryptophan. All right. First situation, we have tryptophan. Ribosome moves fast through there. It gets up over here. And now these sequences are allowed to pair with each other. This guy is a terminator. What's it going to tell the RNA polymerase to do? Stop. When we have abundant tryptophan, it's telling the RNA polymerase, stop. When we have very little tryptophan, the ribosome is sitting right here, covering up these sequences so they can't form. When there's very little tryptophan, the ribosome is waiting, so this guy forms because these guys can't pair with each other. It's blocking it. Now, this to me is amazing. This is an amazing complexity built into something that looks like a very simple structure, and it allows the cell to control, do I make the operon or do I not make the operon? If this can't form, this says, whoa, we're low on tryptophan. The RNA polymerase goes out, and it makes all 10,000 bases of that operon. That only happens when the cell is short on tryptophan. Isn't that cool? You guys say, see, we're too tired for that to be cool. It is cool. I should tell you a joke. Okay? Okay. How many people have ever heard my Crunchbird joke? Okay, don't give away my Crunchbird joke. All right. So this is one of my favorite jokes. All right? A student told me this last year. Blame the student if you don't like this joke, okay? So, Crunchbird. This lady goes out, and it's her husband's birthday, right? She goes to the pet store, and she tells the pet owner, you know, I want to get my husband something different for his birthday this year. I always give him the same old thing. I want to give him something different. Pet shop owner says, I got what you need, okay? He goes back and he brings out this giant anaconda, you know. He gives it to the lady and she says, wow. She says, it's different, but I don't think I want to have an anaconda in the house. Okay, so he takes it back, you know, and he comes back and he's got this big iguana. You know, how about this? This is different. Nah, it's a little big. I don't think that would work in our house. So he goes through a few things, you know, in the same story. No, it's not going to work. It's not going to work. And finally he says, I know what you need. He says, you need the Crunchbird. She says, what's Crunchbird? He says, you see a little bird, that perch over there? She says, yeah. He says, watch this. He said, Crunchbird, chair. Okay. Bird jumps off the perch, flies over this chair, it starts pecking at it, and demolishes the chair in front of her eyes in the matter of a couple of minutes. She said, that's amazing. Little tiny bird. He said, you ain't seen nothing. Crunchbird, desk. There's a great big desk back there. And literally, in 30 seconds, it's sawdust. Bird just goes through it like it's butter. She said, oh, I got to have this for my husband. So she pays him the money, and she takes the bird home. And she gets home, and the husband is sitting there, and he's reading the paper, and he's hungry, and he's grouchy, and so forth. And she says, honey, she says, I got you something for your birthday. What is it? She said, you see this here? She said, yes. She said, this is the Crunchbird. He said, Crunchbird my ass. Okay. I hope that worked for you. It's a cute joke, I think. Okay. Let's finish up. So, any questions about this before I leave the Crunchbird? Okay, yeah. Nope, replication is not involved here at all. So all we're making is, well, replication, we're making RNA, but not DNA replication. Yeah. So we're making RNA. So the question is, do we stop transcription? In other words, over here we stop it. Over here we don't stop it. So if we don't stop it, then the whole operon is made because the cell has gotten the message, we don't have enough tryptophan. We've got to make enzymes that will give us tryptophan.
If it does continue to that anti-terminated,
Yes.
so what happens if it doesn't?
So if it has plenty of tryptophan,
Oh, it has plenty.
see, it stops right here. If it's, and notice we have a U right there. It stops it right there. Okay? Question?